Today is our last day for the module. So before we start, I would like to ask to download a few files. So what I'm going to do, I'm going to share my screen and then show you the files that you need to download. And then we go from there, okay? So if you see my Google Drive type 1, please will put the link to the drive on the chat. Okay, so today we have only a few files to download. So we have RNA-seq annotation files. We have RNA-seq gene expression, have RNA-seq downstream analysis, genomics.zip. I'll tell you what to do with that. And RNA-seq underscore volcano.plot. Okay, the first three files, you downloaded them yesterday. But yesterday, when during the lecture, one of the participants identified an error in one of the files, so I had to go back and correct those files. So when you download these files, these three files, just replace them with the ones you downloaded yesterday. And then these two are new files, the genomic files and RNA-seq volcano. So these are the files that you need to download. Okay. So after downloading these four files, the one with RNA that starts with RNA and -seq will be, you have to put these four files in the directory of RNA-seq that you created yesterday. Okay, I'm going to start again. Okay, you're going to download these files that are highlighted. The 1, 2, 3, 4 files plus one zip file for genomics. That's for our new topic today. After downloading these 4 files plus the zip file, put the RNA, any file that starts with RNA and -seq into your bioinformatics /sec RNA-seq folder. And then after doing that, you unzip the genomic.zip file and then move that file, that folder, to your bioinformatics folder. Okay? So you'll have bioinformatics, and then within the bioinformatics folder, you're going to have genomic folder. And the genomic folder will have different files that I'll go through. So I'm going to give you some few minutes to download these 4 files plus the zip file. And then after that, we'll unzip the genomics file and get the genomics folder and put in our bioinformatics file. And then after that, we'll start our lecture. Okay. So I'm going to give you 2 minutes. And when you're done, you can type 1, then we go from there. Again, I'm going to highlight the file, the 4 files plus the zip file that we are downloading from the Google Drives. These are the 4 files, the 4 RNA-seq file plus genomic zip file. So when you're done downloading, you can type 1 in the chat. Downloading all these 4 files plus the zip file. Okay, so next is I'm going to go back to I'm going to my desktop. I'm going to show you what to do with the genomic file. After unzipping genomic file, you'll have files inside the genomic files. We have population underscore structure, have genomics underscore BC1 underscore BCR analysis, have genomic maze, and genomic package. I don't want you to remove these files from your genomic folder. What I would like you to do is to copy the whole folder and put it in your bioinformatics folder. Okay? I already have mine there. Just move the unzip file and put in your bioinformatics folder. So you should be having your genomics folder inside your bioinformatics folder. Okay? So again, after downloading your genomics.zip file and zip it, like what you did yesterday with RNA-seq file, put it in your bioinformatics folder. We'll come and work with that after finishing our RNA-seq where we left yesterday. Okay? So if you've done that, just type 1. If you've completed, you downloaded the files, the 4 files, RNA-seq files from the Google Drive. You downloaded the genomics zip file. You unzip the genomics zip file and you move the folder to bioinformatics, type 1. Most will not yet. I'll give you another 5 minutes to do that. Okay? So go to the Google Drive. Look at the Google Drive. Download this 1, 2, 3, 4, 5. That's with RNA-seq. And also download the genomics.zip file. And these 4 RNA-seq, put them in your RNA-seq direct folder in your bioinformatics project. And then after that, unzip the genomic folder. After unzipping, it will create for you a folder by itself. And then move that folder. Mine is here. Move that folder to your bioinformatics project folder. Okay? Okay, type 1 if you're done with downloading your files, unzipping the genomics, and then moving it to, let me go back here. Let me show you clean. Okay. Okay. Yesterday, okay, type 1 if you can see my slides. Type 1. You can see my transcript of slides. Type 1. Okay, thank you. So yesterday we started with transcriptomics, what's RNA-seq. We watched two videos. One was about the messenger RNA is made from the nucleus, and then it's exported to the cytoplasm, and then the translation takes place, and then you get your protein. And we also watch a video of RNA-seq. And then after that, we went through the workflow of RNA-seq. Okay, so we started with cleaning, cleaning our FASTQ files. After cleaning, we checked because we had some errors from the machine, the first 15, because the ratio of A to C and C to G, we had some problem, but we didn't have any problem with the threads. All our threads were above 30, so we didn't clean that. Then after cleaning, we checked again. After checking, we found that, okay, now we have good sequences. Then we did alignment. We line our reads. We line our reads to the genome because we're working with the yeast. And our genome, yeast came in a FASTA format. And then after aligning the reads to the genome, we had to find out what are those genes that, what are those genes that were aligned to the reads. So we did the annotation. Basically, we annotated the gene, those that were aligned. Then we got our matrix, the table. Then from there, we did basically visualization to find out if the experiment worked. And we used three methods: use cluster analysis, use principal component analysis, and principal coordinate analysis. So it's always advisable to use at least more than two methods. So the principal component analysis was the best to show that the experiment really worked. So they treated and the checks or the wild type was separated. But that's where we stopped. So what do you do with that? So the next we're going to do, we have two steps left. We're going to do the gene expression analysis. And then after that, we're going to do the downstream analysis. That's where we're going to start from today. Okay? So I will go and start from there. So this is where we stopped yesterday. So today we are going to step 4, the expression analysis. So what's differential gene expression? So it's always written as DGE. It's a method to identify genes with significant expression changes between experimental condition, example, treatment versus control, like ours was treatment versus control, or if you're working with diseases, healthy versus disease. So these are the steps involved. You quantify your expression. That means you count your RNA-seq reads per gene. That's where we stopped yesterday. You normalize, adjust for sequencing depth and library size, and then you do the statistical testing. So there are many methods that we can use, but for us, we're going to use HR. That's why I'll put it in red here. And then after that, you adjust for multiple testing, like control for discovery rate and so forth. And then the last is to generate your results that will show you the upregulated and downregulated genes. So what does that mean? So when you get your results, you'll have some positive and negative. So the positives are the upregulated genes in your treatment. The negatives are downregulated genes in your treatment because they are basically highly expressed in your check or in your wild type. Okay. So after that, then we have some few outputs that you have. You have volcano plots. We watched that yesterday in the video. You have heat maps, and then you have a certain maps called MA map. And then also the adjusted p-value. So the purpose of uh, expression analysis is to identify biological signature. And then after that, you also do functional notation to show you which pathways are involved in your gene expression. Okay. And then after that, so you can also use those to provide your target for validation. Okay, the package that you use is called HR. So what does the package do? So the HR works with a table of reads count. So the rows correspond to your genes and the columns to your independence library. And the counts represent the total number of reads aligned with each gene in the genomic locus. So the input of HR is always the output of future counts. Well, that's why we stopped yesterday. For example, because yesterday we used the package called RSubread. So the output of RSubread is the one that we're going to start running today. So we're going to run, we're going to do gene expression together. But before we do gene expression, we need to start from where we stopped yesterday, because we are doing a RNA-seq workflow. Okay, so I'm going to share with you how we should do it. So this time we'll just we'll go to RStudio, open RStudio, and then from RStudio we're going to be in the RNA-seq directory. We'll do that together, and then we'll start running each script from yesterday and RSub, and then so I'm going to do that. And the script that you're going to run is called RNA-seq underscore gene underscore expression. You downloaded this one today, replace that of yesterday. Okay. So I would like you to go and open your RStudio. Okay, so this, I would like to, if you see my word, please type 1, because we're going to go through this. Please type 1. Thank you. So these are the script we have. The ones that in red is the ones that we covered yesterday. And we are going to do the same. We're going to start with RNA-seq. Oh, no, this one, we are not. We're going to start with number 2, because this number 1 we use for installing packages. We're going to start with number 2. That will bring our sequence. We're going to do the cleaning. We're going to do the check. We're going to do the alignment. Okay, this one you downloaded today because we updated it. And then you're going to do exploratory data analysis. We're going to do filter. And after that, then we'll go to number 9. When we after finish number 9, then we'll do number 10. Okay. So you'll follow me. We're going to do this together. We're going to open our RStudio, and I want you to follow me so that we go to our RNA-seq together, so that when we are running, uh, we are in the same directory. Okay. So I'm going to open my RStudio. So if you can see my RStudio, Studio, please type 1. Okay, I would like you also to open your RStudio Studio and follow me exactly what I'm going to do. So if you don't have your 4 windows, just come here and click on Show All Pane and then adjust accordingly. Okay, I would like to click on File. Okay. So if all of you should click on File. So I would like us all to do this together, everyone, because I would like us all to be in RNA-seq directory. So we're going to do this in a long way, but it will save us a lot of time. So on your right lower window, I would like you to click on your file and then go to your right. There are 3 small dots. Okay. I'd like to click on those 3 dots and then look for your bioinformatics folder and open it. So when you open your bioinformatics folder, you're going to have home, desktop, and bioinformatics. I would like you to, if you see your home, desktop, and bioinformatics, type 1. I'm going to repeat this again, but if you can see that, type 1. Still be in your file. Okay, good. Now, that's good. Now, I would like us again to go back and click on the 3 dots again. Okay? Oops, we missed one step. We missed one step, okay? Uh, uh, let's start again. Let's go to the 3 dots and look for your bioinformatics folder and open it, okay? So if you see your home, desktop, and bioinformatics, okay? Home, desktop, and bioinformatics. Next, you see this kind of like a wheel, that screen. I would like you to click on that and set it as a working directory, okay? You still have your home, desktop, and bioinformatics. But go to this small wheel here, click on that, and setting as a working directory. Okay? Now, we're going to go to another extra step again. Go back again and click on the small. And now, what you're going to look for, click on your bioinformatics and click on your RNA-seq and open. Now you have home, desktop, bioinformatics, RNA-seq. Home, desktop, bioinformatics, RNA-seq. And now go to this small wheel here, click on that, and set this as your working directory. So now we are in our working directory. If you're there, type 1. Okay, I'm going to repeat again. So those ones who have not reached here, I'm sure you have your home, desktop, and bioinformatics. Go back again, the 3 dots here. You click on those 3 dots, look for your bioinformatics folder. And within that bioinformatics folder, there's RNA-seq. You click on that, you open it. And then after open that, you go to this small wheel here. Go to more and set as your working directory. If you can do that, then we are ready to go. Okay? So we're going to follow this. We're going to start with RNA-seq package. We're going to start with, let me remove this not to confuse us. We'll start with RNA-seq quality check. We're going to do cleaning. We're going to check the quality again. We're going to do alignment. We're going to do annotation. We're going to do exploratory data analysis. We're going to do filter. When we have done that, and then we are going to now do what? Or gene expression analysis. So I'm going to go back. So now, since you're now in your RNA-seq folder, you're going to go to your left where there's RStudio, click on File, Open, and look for the first script. Our first script is RNA check. Let me see here. RNA quality check. This one here. RNA-seq quality check. And open that one. Let's see some question here. Let's give the rest at least a minute. Okay. So if you have opened it, what I would like you to do is just to highlight everything and run it. Because we are already in our RNA-seq directory. So we're going to run this. It should be fast. So the product of this is going to go to the next step. Okay. If you have been successful, right, type 1. Okay. Somebody has error. What's the error? Because I don't think you have the file. You have the package. That means your file is empty. Yeah, you did install the package. That means your file is empty. Okay. So those who are getting errors, please install your package. Okay, so the second file that we're going to run is the cleaning file here. We'd like you to run this one here. RNA-seq underscore cleaning. We are not going to check, we're not going to check, we're just going to do the cleaning. Okay? So this is the second file you're going to run. So the first one is quality check. The second one is the cleaning. Okay? So again, just run that. Highlight everything and run it. So we're trying to save time. So yesterday we checked our reads. We saw where we had problems. I did not run that because we know where the problem was. We're also going to skip another script. That script that showed us that our file was clean. We're going to skip that. Now, since we have a clean file, we're just going to go and do alignment straight. So I'm skipping two scripts. One was for us to see how our sequence looked like. I've skipped that because we did that yesterday. So I got files. I got the files from the computer. I clean it. Now we're going to do the alignment. Okay. So the next file we're going to work is to run the alignment file. This one here. Okay. So we clean our file. Now we're going to do the alignment. Because we did all this yesterday. But what we're going to do today, we cannot do without the products of what we have been doing. So we're going to run this. Okay. I'm going to go back to our script. I mean, to the Word file. So we did this one. I'm going to change the color here. Blue. We did this one. We skipped this one because yesterday we already did that. When we did the cleaning, we skipped this one. We did the alignment. Now we need to do the annotation. So we need to do the annotation. And then after annotation, we're going to, I'm going to skip exploratory load analysis because of time. That one we saw our PCA and cluster analysis and those of yesterday, because the product of this fed to this, the product of this fed to this, the product of this is going to feed to this. So after that, then we can do our, we're going to skip this, then we do the normalization, and then we will start our analysis for today. So we have 2 runs left. We're going to do annotation and normalization. We're going to skip this script here because yesterday we saw what he did. So the product of this will fed in this, and then we'll do our analysis, okay? See that here. So if you're installing and it says update, please click A and run that, okay? Okay, so I'm going to go back to our ARA Studio. Next, we're going to do annotation, okay? This is the annotation file. I'm going to open that. I'm going to run it. And then after annotation, we're going to do normalization. So annotation, next we're going to do normalization. This normalization file here. Filter is filter normalization. We're going to run that. And that will give us our matrix. This is where we stopped yesterday. So now next we're going to do the gene expression analysis. So this is new. So you're going to go open your file, and this is the script that we need today, RNA-seq gene expression. I'm going to open that, and I'm going to run it, and then after that, I'll go back to the slide and show you what happened. Okay. So we have done our gene expression analysis. So I'm going to share this with you so that you can let it be running in the background when I'm explaining on the slide. So we ran this, we ran this, we ran 4, we ran 5, we ran 7, and we ran 8. So I'm going to put it blue. Okay. So now I'm going to go and explain what were the results. If you see my slide type 1, okay. So basically, this is what's going to be our output. We'll see that later. So the output will have different columns. The first one is going to be gene ID. That will tell you the names of the genes in yeast. And then it's going to be log fold change. Greater than 0 means the treatment, the genes are upregulated, and less than 0, that means the genes were downregulated. And you're going to have your p-values and fault discovery rate. We're also going to have log of count per million, but we won't care about that. And then also the results are going to be saved in the folder, okay? So let me go through this, explain when you look at those columns of p-values and fault discovery rate. So p-value tells you that you're willing to accept 5% of all the tests to be false positive. So false positive means that if the gene was supposed to be upregulated in treatment and it was downregulated, it will tell you that it's upregulated, then that's false positive. And then after getting those, assume you had 200 genes that were upregulated. You need only 5% to be wrong. So 5% of those will be like maybe around 40. And then you'll be left with 95%. So the false discovery rate now, you go to those 95%, and then you you also can imply that only 5% of your p-values, you can live with that. So the false discovery rate is kind of a little bit conservative. And in RNA-seq, they use false discovery rate because of the number of genes, because we have thousands of number of genes. So this is the adjusted p-value, okay? So when you run your script, it saves your data in a folder. It creates a folder called DE_results. And then when you open that, you find what's called prepare. That means comparing treatment one, and then treatment one, and then treatment one, and then treatment wild type, and treatment. And then outside, it's just comparing all the treatments because our experiment was treatment 1, 2, and 3, and then wild type 1, 2, and 3. So this pair is comparing those. But outside, you have all this. That's comparing the whole, combining treatment 1, 2, 3 together and wild type 1, 2, 3 together. And then now when you open this pair here, you get now these are the comparison here, okay? So when you open one of those files, you have gene ID. So this is how the genes are named in yeast. So these are your gene IDs. This is your log fold change. So this gene here, it was upregulated in the treatment, okay? And then this is log count per million. We don't worry about this. And this is log ratio. We don't worry about this. You can either use p-value or false discovery rate, but we always use false discovery rate because it's very, very conservative. So there are 4 columns that you have to pay attention to. The first one is gene ID, will tell you which gene was either upregulated or downregulated. So gene number 1 was upregulated in the treatment. Gene number 2 was downregulated because it's negative. So that's how you read your results of gene expression analysis. And we also have they always cut point of plus or minus 2. But if you find it very stringent, you can go to plus or minus 1.5, okay? So the first gene was upregulated in treatment, the second gene was downregulated, third was downregulated, and so forth. So this is how you get your results after going through all this. It's how you get your results. Okay? Let's have some questions here. Someone said change the directory, but the problem is file name object does not contain any value, because when you change the directory, then you're out of the system, the error system completely. That's why we went through and all of what are supposed to be in RNA-seq. So when you change the directory, that means R will not read your files really. Okay, the next thing that we can do the volcano plot, like that we watched yesterday. So the volcano plot will tell you on the x-axis is your log of p-values, or on the y is your log fault change. So any dots that are black, those are not significant. On the left, they're down-regulated. On the right, they're upregulated. So we have a volcano plot script that we are going to run later. Okay? So this is a new script for today. Okay? So let's go back and run the volcano plot. After that, I'll come back and continue, and then we'll do the downstream analysis. So we're going to go back to our ARA Studio. Okay? So you can, if you see my ARA Studio, type 1. Okay, thank you. So next we are going, there's a script called volcano plot that we got today. So we're going to open that, this script here. And when you run that one, you have to set this to a pair because it was saved there. So when you start running from from the line 1, you should be able to get this plot here. Okay? So now from when you did your experiment in the lab, you sent the samples for sequencing, you came and ran everything. Now you have your genes of interest. Some are upregulated, some are downregulated. What do you do with those genes? Okay? What do you do with those genes? So this is going to bring us to our last part of RNA-seq today, before we go to genomics. Okay, so at the end of every experiment, you want to find something that you're going to use. I still go back to the maze. So assume our treatment. Let's forget we are not working with yeast. Let's assume we're working with maize. So I'm a breeder. I went to my field because of climate change. It didn't rain, but few plants survived. So those few plants are going to be my treatment. Okay, those ones that died are going to be my check. So I'll go collect those, get the samples from them, extract the RNA, send the RNA for sequencing. The sequencing will come back and we'll do the same process that we did for yeast. And then at the end, we're going to get the genes for maize will be on the left. And then you have your fault change. So like this one here. So the gene ID here, this will be now for maize. Okay. And then let's assume number 1 was one of the gene for maize. And then the log fault chain will be for treatment. So this gene here will be upregulated in one of those maize that survive. And this gene here, if assume this was maize, will be downregulated in in the maize that survive. So we'll be interested to find out, okay, what are these genes doing? So now you can apply your RNA-seq in plant breeding. But stopping here alone won't help. You've done your best. You got these two groups. Some died because of heat and some survived. So assume this was your maize genes. That number 1 was upregulated. Number 2 was downregulated. What can we do now? Now you go and do the downstream analysis and find out what are these genes doing. Okay. To do that, you use what's called gene set pathway analysis. So we did number 4. We got the genes that were expressed. Some were upregulated, some were downregulated. This is the last step here. It's called functional annotation. You can use KEGG, which you're going to see. You can use GO, you can use string. Okay. So you can also draw heat map, but we'll do that. You can also draw heat map. Okay. But this is just heat map show. This is the heat map to show you maybe the top 20 genes. So the functional notation. So what do you do? The functional notation is the process of assigning biological meaning to those genes that are expressed. So how do you do it? It involves identifying those genes, mapping them to the databases. That's why bioinformatics databases are very, very important. You can do everything, but you still go back to those databases. So you match those IDs to known biological resources. You have gene ontology, you have KEGG, you have Interpro, and the rest. So they will tell you that your gene that was upregulated does this function, or your gene that was downregulated does this function. Okay? So the purpose of functional notation is to reveal for you biological process driving the changes. So what does that mean? So you have heat, the climate change, but the genes that were in those plants that were expressed, they were able to withstand those heat. So how do you find that? You have to do functional notation. Also, it tells you the pathway that they're involved in, and you can now use that for your biomarkers. So in the future, when you plant your maize, you can now use those genes to do selection. And of course, it can also support your hypothesis generation, okay? So I'm going to go briefly, since I'm going to share with you these slides. In your free time, you can go and look at all these websites. The first one is gene ontology. So the mission of them is to develop a comprehensive computational model of biological organism. So what they do, they tell you, they have three things that they tell you about the location of that gene, the molecular process, and the molecular function, okay? So because of the time constraint, I won't go to the website. So that's what gene ontology does. So when we send you these notes, you can go and read more. Another one is KEGG. KEGG stands for Kyoto Incorporated Genes and Genomes. So it's a database of understanding eye function of your genes. So basically, it does work with enzyme, other molecules. So again, we have the website that you go and find out more about the KEGG. Interpro. Interpro is a website that classifies your proteins. It puts them in different domains because the domain of the protein also help you to see what is going on. So this is one of the databases that you can use it for your functional annotation. Another one is PFAM. So this PFAM is a collection of protein families. So it represents multiple sequence alignment. We covered that. I also use hidden Markov model. So again, we have the website for that. So in your free time, you can go and work with that, okay? Last but not least is string. String is a database that shows the protein-protein interaction because for these maize to survive, they have to interact with other proteins, okay? So they get their information from genome content prediction, therapeutic lab experiment, and so forth. So this one, we also have the link to those to the database. Okay. So we are going to run our last script for downstream analysis. And it's going to create for us a folder called string results. And these folders will have gene annotation, biological process, cellular component, molecular function, and also it will also give you the KEGG and other things. But to run this, you have to be connected to the internet because the script go to all these databases and bring and compare with our express genes. Okay? So let me go back to our other studio. Okay? So our last script for the annotation is going to use the output from the gene expression. We're going to go to open. We're going to go to RNA-seq, and the script we're going to get is the annotation script. Let me see if I have it here. No, it's a downstream, sorry. Downstream analysis script. It's this one here. So if you run this script, it's going to be a little bit slower because it depends on the on the speed of your internet. So what's going to do? Error. Let me start here. Why am I getting error? With this one, I think we should go line by line. Let me start from up here and see why it's giving me an error here. Okay, I'm going to start line by line here. DH certificate size not found. Something is going on here. Let me see. Don't I have the package? I ran this and it worked. Okay, let me do something different here. Let's see when. Okay, I have some problem here. So I will work on this because it ran. So what I'm going to do, I'm going to show you the results, what I got, something happened. So to save time, I will work on this tonight. I will send you the script. Something happened here, but before the class mine ran, and these are the results, okay? Let's save time here. So, mm, Richard, relax. Pardon a sec. Do you see my screen? If you can see my screen, type 1. So this is what I got when I ran the script. So we're going to go through this. Something happens, it's not running now, but I'm not going to spend, we need to do genomics. So I'll start with this file here. So this is my annotation file, okay? So if you look at this, do you see my Excel file? Type 1. Okay. So these are my genes. And this is the first default discovery rate. That's what I was using. And this is the one that was upregulated. And this is the gene symbol that's annotated. And the description is it's 3-phosphor. So it's kind of a long gene here. So it tells me what this gene is doing. So now I can go back and read more about this gene. So this gene, if this was maize, this gene was upregulated in my green maize that survived. And I go to the second gene, assume this was maize, this gene was suppressed, downregulated. I'll just give it a blue color. And this is the name of the gene. So this is my annotated file. So now I've started now making sense with all those genes, okay? So that is part of the annotation. So the next is the gene ontology. So what are the processes going on here? So this is processes. So there were 11 genes here that belong to this, and they match to that. So this is, the process is cytoplasmic translation, and this one is translation, and so forth. So this will now give me the process. And when I go back again to the gene ontology, the cellular components, where are these genes located? You can see some of them are located in ribosomes, some are located in cytoplasmic ribosome, and so forth. And then now I go to the molecular function of those genes. You can see that some of them are structural and so forth. Some are molecular activity. So that is from gene ontology. Now I go to KEGG. So KEGG only had what? Only had one hit. And then now I'll go to and look at this, the term, I go back to where? I go back to the KEGG website and I go and look at this. What does this do? And then these are the genes that are interconnected with that. Okay, if I go to string, these are the genes that are interacting. See that? And so forth. So I still have this, the top one. And then now if I look at the map, it's a string map here. These are the genes that's how they're interacting. So the functional notation will give you all this information. So in the process, you can get the markers from them. In the process, you can go and maybe do some experiment in that. So because of time constraint, I'm going to stop here. I'm going to do genomics. I have to do genomics because today is our last day. Okay? So you'll forgive me for that. I will send you the correct script for downstream analysis because I ran it, but something happened here. So forgive me for that. I have to go and do genomics because it's very, very important for this group here. So let's go and start genomics. It's our last topic. And then, okay, before we close, we're going to do post survey. So if you can see my slide type 1. Okay, thank you. So our last topic is genomic data analysis. Okay. So what's genomics? Genomics is a comprehensive study of all organism DNA genome. So when we watched the video, the first video yesterday, we were told that human being has 23 pair of chromosomes. And within the chromosomes, you have this DNA that go around the histones. So everything there is the genome. It's a genomics. So it includes its genes, their functions, and the interaction. So it focuses on structure, function, evolution, and mapping of all of the genomes. It goes beyond single gene. Yesterday, when we watched the first video, that was one gene was switched on. After being switched on, it was translated with messenger RNA. The messenger and RNA went. That was one gene. But genomics is dealing with everything. So you use advanced tools like sequencing, bioinformatics, statistics, and AI. So why are we interested in genomics? I've been talking about maize. It helps crop resilience in varieties, accelerate breeding, and it enhances livestock genetics, support precision farming in agriculture. Medicine, it enables personal treatment, improve the early disease prediction and prevention, support pharmacogenomics, ensuring that drugs are effective for specific patients, is also used in advanced cancer, in identifying tumors and so forth. We also have in public health. It's used for disease surveillance. So sometimes when they say an outbreak, they go and do the sequencing and they use genomics to track this change in pathogens. Monitor antimicrobial resistance. Use genomics, guide development of vaccine, diagnostic, and so forth. Okay? Okay. Like in metagenomics, in RNA-seq, there are also different types of what we do in genomics. One is whole genome sequencing. So this is a laboratory method that uses to determine the complete DNA sequence of an organism in a single time. So what you do, it covers all coding and non-coding regions of the genome. So if you have the genome, you have all this DNA, some parts are codes for proteins, some don't. Okay? And he uses high throughput sequencing technology like Illumina, Oxford Nanopore, and provide base-to-base view of genetic variation. So when you're trying to sequence a genome for an organism, you use this whole genome sequencing. Okay? So what's the advantage? You do what? You sequence the entire genome. It has very high resolution, detecting SNPs, indel, and also enables comparative and functional genomics. What's the limitation? It's like when we were doing shotgun, it's very, very expensive. It requires very strong bioinformatics capacity. Okay? So this is the workflow. You get your DNA, you do the extraction, you prepare your libraries, you use the sequencer, can be Illumina, PacBio, or Nanopore. You do data processing, base calling, we'll see that in a minute. You do your alignment, you do assembly, you can do variant calling, you get your SNPs, and after that, you do your downstream analysis. The next one is called genotype-based sequencing, GBS. So GBS, you don't use the whole genome. You use restricted enzyme to cut those into small chunks. Okay? So you're not using the whole genome, you're just using restricted enzyme. So it makes it a little bit easier and it's cheaper. You generate your SNP. So sometimes you start with that so that it help you to complete a reference genome when when you just want to fill the gap. It's not very, very expensive. Okay? What's the advantage? It's cheap. You can whole genome. You can do rapid genotyping of large population, effective for non-model species. Limitations, not comprehensive enough, missing data and even coverage, lower resolution than whole genome sequencing. This is the workflow. You get your DNA, do the extraction, get your restriction enzyme, do the digestion, you get your adapter, you do the PCR amplification, you send it to sequencing, you do your data processing, you do the SNP calling, and do your downstream analysis. So if you compare the two, start from the left, genome coverage. The whole genome sequencing, you do the entire genome. The GBS, you do a subset of it. The output, very high, billion of reads. It's moderate selected fragments. The cost, very high to do WGS, very low to do GBS. Resolution, detect all the variants, including SNP, indel, and structural variants. This is mostly used in SNPs. Restricted enzyme, WGS is not required. This is required, and this is best for detailed genomics and rare mutation discovery, many used in medical field, and this is basically used for population genetics. Okay? So this now brings us to genome-wide association study. So this is used to identify genetic variation that's associated with specific trait for the entire genome. So it examines millions of genetic variants, especially the single nucleotide polymorphism. We're going to talk about that. It's population-based, compares the DNA with individuals of a particular trait or disease, and then you use statistical association by testing each SNP or GWAS, finding the variants that occur in individuals. The good thing is there's no prior hypothesis needed for this when you're doing GWAS. Okay? So what's the advantage? It's unbiased discovery. You can get new genetic association, can reveal biological pathway, works well with complex traits by many genes. Limitation, large sample sizes to detect small genetic variation. Association also sometimes does not prove causation. They only analyze candidate regions. Many significant variants are in non-coding regions, so it makes biological interpretation harder. Why? Because biological interpretation mainly come from the coding region. That's where you transcribe your messenger RNA, and then it gets out to be translated into protein. Okay? So GWAS versus GBS. Definition, statistical approach to link genetic variants with trace or disease across the whole genome. GVS, sequence-based method to discover genotype SNPs by sequence and reduce repetition of the genome. The primary purpose, identify association between genetic variants and phenotype traits. And the primary purpose for GBS, to generate your SNP, and eventually it's used for diversity, QTL matching, or breeding. The input requires, for GWAS, sometimes you require your SNP arrays, GBS sequence plus phenotype data. For GBS, you just require data to generate your sequencing. The output, statistical association, Manhattan plots showing SNPs are linked to traits. The output for GPS is only SNPs across population. Okay. Okay, what are the applications? The GWAS, human disease genetics, agriculture trait in mapping, population for GBS, plant breeding, diversity study, and so forth. Advantages, unbiased discovery of genetic loci, cost-effective, work for species without reference genome, high density. Limitation, needs large sample size. The other, for GBS, missing data because you're not covering the whole genome. Relationships, uses genotyping data from methods like GBS, SNPs array, and WGS. Provide genetic data that can be used for GWAS. So basically, when you start GBS, will give you data that can be used for GWAS. Okay, how do you process genomic data? Always start with raw reads, FASTQ file. Do the cleaning, quality control, you do the reading mapping, you align the reads to reference genome, select the best hit from multi-map reads, you do the BAM conversion, then you do your SNP variant, okay? And then you do downstream analysis. Okay, I'm going to stop here a little bit, and then I'm going to look at what the type of question they're asking, then I'll come here. Where do I get a PDF of this PowerPoint? Okay, the PDF, we always put them after each webinar on Google Drive. Okay, I don't think I have many questions here. So, okay. So, this is where we're going to start doing the analysis. So, when you're doing RNA-seq, you're doing metagenomic, you're doing genomics. They all have to be sequenced by what's called high throughput. And this high throughput means that you're sequencing just in bulk. So you prepare your library, you send your sample, you get your FASTQ files, you did your quality control, and you did your alignment. So what we did yesterday in RNA-seq is the same thing when you get your data for genomics, it's the same thing up to alignment. So the script we use up to alignment is the same script when you're doing genomics. The only difference is when you're doing, instead of using RNA somewhere, you use DNA. But it's the same process. You check for the DNA, you check for Fred, you check for the nucleotide ratio, you check for adapters, you go through the same process until you reach alignment. After alignment, that's where now they deviate. In genomic, after alignment, we do what's called SNP calling. And after doing SNP calling, you get what's called VCF file. We'll talk about that later. Variant call file. And these variant call files are the ones now you can do your association study, that's always GWAS. You can do your genetic diversity study or population structure study. Okay, so that's why I put here first to show you that the process is always the same until you reach alignment. So this is what we did yesterday. We got your library, we got our first Q sequence. We did the quality control. We did the alignment. We aligned the reads to yeast reference, and yeast reference genome was FA. And then after alignment, we had to assign, the genes. We use reference annotation file that was GFF3. And after that, we counted for every gene, how many counts, how many reads were mapped. Well, then we got the matrix. That's where we stopped yesterday. And then when we came to started, we went and did gene expression to show which ones were upregulated and which one were downregulated. And then after that, we went and did functional notation. We did gene ontology. We did KEGG. So this is the process of transcriptomic or RNA-seq. Okay. Now, when we go back to genomics, after your alignment, you have to get SNP. We're going to see what SNPs is. And then after getting your SNPs, you're going to get your variant call file, your VCF files. And on those VCF files, you can either do GWAS, association, you can do genetic diversity study or population structure, which we're going to do here. Okay? But before we go, let's talk about what's single nucleotide polymorphism. How do you identify it? So single nucleotide polymorphism, SNP, always pronounced as SNP, it's a variation in a single nucleotide, ATCG. It's a specific position in a DNA sequence among individuals or species. For example, this is a normal DNA sequence, ATGCAT. And this is the variant. A is still the same, but you can see T is represented by C. G is the same, C is the same, A is the same as T the same. So here, T is substituted with C. So the change is a SNP, which is a single nucleotide. We have 1, 2, 3, 4, 5, 6 nucleotides. This is the normal sequence. But the variant, A is still the same. T has been substituted with a C. And when it happens in a coding sequence, it can change everything. It can change everything because remember when we're doing translation, every 3 codons represent an amino acid. So assume ATG, I think this is methionine. I think assume ATG is an amino acid. Okay. And then when this is represented, substituted with this, you're going to get a different amino acids, and that might change the protein completely. So if you go back to our our maize, maybe in that specific gene, the ones that were maybe normal, they had been do anything. But this one, so the future, the most common type of genetic variation in the genome, so must be present at least 1% of population to be considered SNP. And it occur about once every 300 base pairs in human. Okay? So we have the coding SNPs. They code in the protein where you can, where you can change protein sequence. You have non-coding SNPs and you have silent SNPs. Okay. I'm going to show this with this cartoon here. So you have these SNPs. You have this from 80, everything is the same, but there's a change here. Okay. So let's assume this is recorded, these SNPs occurred in a coding region. So because of this change, because of this, I think this is normal. So the color of this fish is going to be blue. But the variant, because there was a variant here, this fish is going to be orange because assume this is the gene for color. So this protein is a normal protein. It makes a blue fish, but this protein is not normal. It has changed. So this gene now, instead of making a blue fish, it's making an orange fish. So that is a nucleotide, a single nucleotide polymorphism. It can make a big difference. Okay? Okay, so how do we do genomic analysis using RStudio? Okay. So this part is the same like what we did the last 2 days. And then when you do alignment, when you're align with the reference genome, you're going to have either SAM or BAM. As I mentioned yesterday, SAM is easy to deal with. I mean, BAM is easy to deal with because it is a binary format of SAM. And then you do your SNP calling, and then you can do this, okay? So I did some work with my colleague at Ohio State with backcross in rice, but he gave me permission to use the variant call, not the raw data. So I'm going to skip this. I'm going to skip this part here, the SNP calling, because we did the SNP calling, but I'm going to give to demonstrate with the results of after SNP calling. So we had backcross 1 and backcross 2 for rice, and I'm going to explain what the backcross is. So is a rice molecular breeder. We work together. So working together, so we sequence after growing rice, we align to rice genome. We did the SNP calling, and then we got our variant, our VCF files. Okay, so I'm going to demonstrate the difference between the backcross 1 and backcross 2. Okay, and we are going also to work on that together. We're not going to do the SNP calling, but we're going to pick up from there. So what's SNP calling? Since we're not going to do, but I would like to explain what a SNP calling is. So SNP calling basically is identifying single base change A to T, G to C in a DNA sequence compared to the reference genome. You have to have a reference genome. So what's the process? You sequence the DNA to generate the short reads, like what we covered in metagenomics and RNA-seq. You align your reads to reference genome, like what we did yesterday. And then from there, now you deviate. You start doing variant detection. So you detect, going through all the genome where there was an alignment, and see if is there any difference with the reference genome. Okay. And then of course you do the filtering. There's some statistics that you do, and then your output file is always called variant call file. Okay. And that file stores the position, the reference of an allele, and give you the quality score. Okay. So the application, population genetic study, we're going to do this here. Association study, we're going to do an example here, and so forth. So what are the tools that I use? There's GATK, some tools, Freebase, and we're going to use ARA and ARA Studio. Okay, before we go, I'm going to discuss what is this VCF file. It's called variant call format file. It's a standardized text format for storing your genetic variant SNP. It can store your SNPs, can store your indel, can store your structural variant. So the header starts with this hash, gives you a reference genome, the filters and annotation, and also have the chromosome part, and then the data is a line. So this is an example. It gives number one is the chromosome 1, the position of that chromosome, and this is the SNP ID. So this SNP ID is RS12345, just an example. And the reference genome, this is the reference genome. This is the SNP, and this is A, and then it tells you whether it's a pass or it's not a pass. Okay. So we're going also to use VCF file. We're going to use VCF file. We're not going to start from the reads file like what we did for RNA-seq. We already know the process. So the only thing that we're going to skip is the SNP calling part, because first of all, it's taken a long time, and then I was not able to get the permission to use it. So let's go and talk about this BC1 and BC2 SNPs. So what's a backcross for people who are not breeders? So a backcross is when you cross the parents, and then you get their offspring. And then when you get your offspring, you cross one of the offspring to the parents. Okay. So the objective of my colleague was to identify the SNPs from backcross 1 and backcross 2 generation of the rice. So what's that? What a backcross is? You cross the parents, and then you get the offspring, and you cross them back. Okay. So in your genomic data, genomic folder that you downloaded today, we have three type of files. We'll go there. We have a genome for rice, have a transcript. This that's a notation file, and you have the variant calls. So these two files came from what? These two files came from SNP calling. So what I would like us to do, I would like us to go, we're going to do that together. I would like us to go back to our genomic folder that we downloaded. We're going to pick the folder with all the files inside. We're going to put that in our bioinformatics folder. And that from there, then we're going to start now doing a real analysis of genomics, okay? So I'm going to stop here. If you can see my folder type 1, my desktop, I mean type 1. Okay. Okay, good. So Assume you downloaded the zip file from the Google Drive. I'm going to open that file. That file has a population structure analysis R script, have genome backcross 1 and backcross 2 R script. It has analysis for maize gene GWAS. So we're going to do backcross, we're going to do population structure, and we're going to do GWAS. And also it has genomic package. So we're going to install that genomic package together. And then it has a VCF file, a code variant file for backcross 1, which is backcross 2 and backcross 1. It has a population data, a variant file, and then also it has a RDS file. And then it has the rice genome and also has the rice transcript. So what we were going to do first, if you haven't done it yet, is to move this and put it in your bioinformatics folder. I'm not going to do that because I'm already there. So those who have done that, please type 1. Those who already have that genomic folder in bioinformatics, please type 1. Okay, good. So now we're going to go and open our R Studio. And we are going now to move from our RNA-seq folder, we are going to go into our genomic folder. So if you see my R Studio type 1, okay, so this is what we are going to do together. We're going to close all the script of RNA-seq, close all of them. Close all those scripts, all of them, close them, all of them. We're going to close them. That's the first thing we're going to do. So all of your left windows, the script window should be now empty. We're going to close those. Next, we're going to clean our console. How do we do that? You go to Edit, Clear Console, because I want us to have a clean slate. The first thing we're going to do on our script editor, we're going to delete all the RNA-seq scripts. We're going to delete all of them. Okay? The second thing that we're going to do, we're going to clean our console. To do that, we're going to go to Edit, Clear Console. So now we have two clean slate on the left. Then we're going to go to our environment. We're going to clean our environment. You see this small broom here? Make sure your cursor is on the environment. You click on that broom and say yes, and everything will go. So we don't need anything to do with RNA-seq. So we have cleaned our script editor. We have cleaned our console. We have cleaned our environment. Okay? So if you've done what I've done, I'm going to repeat it again. If you've done what we've done, type 1. If you haven't done, type 0. Okay. So for those who type 0, you have all your RNA script like here. So the only thing I have to do, just click on cancel. You'll cancel everything, and this will be nothing. Yeah, that's the first step. The second step, we're going to clean our console. We're going to go to Edit, Clear Console. Okay. And then after that, we're going to clean our environment. You see this? Make your cursor be on environment, and there's a broom here. You click on that and say yes. So the last but not least, come down and click on Home. You see where? Click on Home. that house there and click on it. That will take you back home. So we have a clean script editor, we have a clean console, we have a clean environment, and we are home. So now we're going to go and get back our genomics. We're going to be, we're going to work in genomics. We want it to be our folder. So the first thing we are going to do, go again to those three small dots, look for your bioinformatics and open it. So now you have your home desktop bioinformatics, and then go to this wheel again and set as a working directory. Okay, I'm going to start again. After clearing all these three windows, there's nothing there. You click on home. Okay. When you're home, go to the three dots, look for your bioinformatics, open it. Then you have home desktop bioinformatics, and come here and set as your working directory. Okay. So now you have home desktop bioinformatics. Now again, go to these those three dots. Now go to your bioinformatics and look for your genomics file. Where is genomics? Look for your genomics. Open your genomics, and again, come to this small wheel and set your working directory. So now you should have home, desktop, bioinformatics, and genomics. Okay? And with all the files inside. Mine, I don't have it because I have very many files. I don't want to confuse you. So you should have home, desktop, bioinformatics, and genomics with all those files inside. If you have that, type 1. If you have your home, desktop, bioinformatics, and genomics, type 1. Okay, so some people say, let's wait for somebody that has 0. I'm going to repeat again. So you have your home. The first thing I have to do is go to look for your bioinformatics, open, and set it as your working directory. Okay? After that, go again to the three dots, look for your bioinformatics, and look for your genomic folder, open it, and set it as your working directory. So when you are done with that, we need to install the software, the packages. So go to open. You'll find the package, and mine is in desktop here. Open this genomic package. If you go and open genomic package, you should see something like this. There are not very many to install. You should see something like this. You don't have many packages today. If you can see that type 1, someone said do not have home in RS Studio. You should, anybody who has RS Studio should have that home here, that house with a home. Okay, so what we're going to do, we're going to run this like we did yesterday, one by one. Okay? So we're going to run this one by one. Again, what you ask, you type A, enter. Okay, because I already installed them. So for mine, okay, there is an error here. Number 9, replace that capital with a small I. Okay, It's replace that capital with a small I on line 9. So if so, line 9 is capital right now, but replace that capital with small I. And you should be able, if after installing, you should be able to see that the downloaded binary package and so forth and so forth. You should be able to see that. Okay. Set my directory. You should be able to see these files. So if you have installed your packages, finish installing type 1. Okay. Okay. So now we're going to do our first analysis for that back cross, okay? We're going to go open this one here. And then you're going to run the library. We're going to read the files. What we're going to do is we're going to compare the SNPs in backcross 1 and backcross 2. Just run the whole script, and then I'll go and explain. I'm going to explain in the I'll explain in the slide. So basically, what we did, we're trying to compare. In backcross 1, we have SNPs that are unique to backcross 1. We have scripts that are SNPs that are unique to backcross 2, and also we have SNPs that are common in backcross 2. So our goal was to only to deal with only those SNPs that are different in backcross, unique to backcross 1, also unique to backcross 2. So okay, you remember I told you to replace that line 9, replace the capital I with a small I. That's why you're getting get that error. Line 9, when you're installing, replace that capital I with a small I, with a small letter I, then it will install. How can I clear the file on the, okay, so to clear your environment, which I'm not going to do, you use this small broom here. And to clear your console, you go to Edit, Clear Console. Then it will go, because if I clear my environment, everything will disappear. We cleared environment because we wanted to get rid of all the RNA-seq analysis. Okay? So let's go and explain the difference now. So if you can see my slide is type 1. Okay, thank you. So these are the SNPs that are common to both backcross 1 and backcross 2. So to be significant, uh, of course, you have to have, they still use the Fred score. So this is chromosome 1, this position of that chromosome in rice. The reference, that's the parent position, was a G, the SNP is an A, and these are the quality scores. And in chromosome 1 again, at this position here, in both of them, in both backcross 1 and backcross 2, the reference, the parents was T, and the SNP was C. But It's okay because sometimes uh, you expect that to happen. But for our markers, we're interested in which SNPs are unique to backcross 1 or backcross 2. So this is the unique SNPs for backcross 1. So in this chromosome position, in this chromosome 1 and this position, the rhythm genome was G, the parent, and alternative was A. And this is chromosome 1, the same thing. So this is maybe that's why some of these SNPs, and that's why we have differences in backcross 1 and backcross 2. This is for backcross 2. At this position, for backcross 1, this position, at this position, the backcross 1 is the same as a reference genome. But with backcross 2, it's different, okay? And so forth. So this is how you can use genomics for identifying markers. So we were able to get markers that were unique to backcross 1 and unique to backcross 2, okay? So this I'm going to show you where when you run your this R script, it's going to store it in your genomic folder. Okay. So that was um, SNP identification. So what we wanted to do was uh, my colleague is a rice molecular breeder. It's a rice molecular breeder. He want to find out, he crossed two parents. He crossed parent 1 and parent 2. He had this trait of interest, and then he did backcross 1 and backcross 2 to those parents. And the backcross 1 is totally different. Backcross 1 has certain characteristics that are different from backcross 2. So we want to find out what are the SNPs that are unique to those. So we did the same thing from here. We did the SNP calling, and then we got our variance code, we compared them. Okay, that's I'm going to show you from this VCF file how to do GWAS, Genome-Wide Association Study. Okay, and then after this, then we will do our we'll do a post survey, but I would like to cover GWAS, and then after that we have two left: genetic diversity and population structure. Okay, how do you associate SNP with phenotypes? So we're going to use data that this work was done and published by European Union. So what they did, they had 256 maize hybrid. This was grown in 2 water regimes, irrigated and rain-fed, in 7 fields in 2013 and 2014. So they collected information on grain yield and its components, and and so forth. So the main purpose of this was to using the environment to characterize genetic variability. Okay. So they published this, and you can get this the RDS file. So the purpose was to determine the allele and correlate them to the what to the traits. Okay, so what they did, somebody I think 2 days ago asked, well, how do you make your packages? So they had to make this package to analyze their data. It's called StatGen GWAS. So you already installed this package. Okay, so this package, I got the data, and after the day after you're going to get the data in RDS, and the data is called GData. It has marker, it has a map, it has phenotype, it has kinship and other covariates. Okay. So the marker has like 39,000, almost 40,000 SNPs, 246 genotypes, and the map has the SNP position and so forth. Okay. So these are the phenotype that they collected. And that's like 13 fields. So they had this was the genotype. Okay, and then they collected grain yield, grain number, seed size, and so forth. This is just typical experiment that people do. So this is the map. So the map, it's not the SNPs, the chromosome, because they already have the map of the SNP and chromosome. So this chromosome 1, the position of this SNP is the position of the SNP. So these are the SNPs. This is the position of the chromosome, okay? So these are the information on the map, okay? So these are the markers. So these are the SNPs you have. These are the information that they collected, okay? So what we're going to do, we're going to run a single trait analysis with that data, okay? So what's single trait analysis? So it's a GWAS analysis where you test the association between genetic markers, which use a SNP, across the genome with one specific trait. For example, you can do plant height, you can do grain yield, you can do this. So this we're going to do single trait analysis. So they gave us the SNPs. They went through the process, they did SNP calling, they collected data in the field, and so forth, okay? So this is their phenotype. They have this phenotype, okay? So they have the phenotypes I showed you. They had the markers, they had the maps, okay? Okay, so what we're going to do, we're going to run this together, and then we'll see the results, okay? And then after that, we'll get some graphs, okay? So we're going to go back to our R Studio. We're going to go back to our studio. We're going to open this genome maze GWAS-R. Oops, 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 oops. Hmm. Whoa. Let me see if this is correct. A disaster. Whoa. Okay. It looks like my script got corrupted. Let me see if I can use the one here. So you just follow my script, okay? It seems like my script got corrupted somehow. Let me do this. Start with library first. You follow what I'm doing, okay? And then I'll explain exactly what's going on here. Bear with me. Let's go single, this one. Okay, this is what I'm gonna do. So if I finish running, then I'll send you the script. And then we'll run it. So okay, so what I'm gonna do, because the other was corrupted. I'm going to upload this, and then you have to download that and run it. So I'm gonna upload that in our okay, you go back to the Google Drive and download that. The maze, this here, the maze, genome maze JWAS here, and run this. Basically, you run it, and then I'll explain what's going on, and then we'll do our post survey. So meanwhile, you're downloading, I'll explain what's going on here. So this is what happened. We're trying to associate the SNP with the grain yield, okay? So if you look at this is the straight grain yield, and this is the SNP that associated to that grain yield. This is on chromosome 6, and this is the position, and this is the allele frequency, this is the p-value, and this is load score, which is the log of the odd, and this is the SNP status, and this is the proportion of the SNP, okay? So when you run this, basically what we are trying to associate the SNP with the with the maize yield, okay? That is the GWAS. And then how do you know that your analysis is good? We do what we call QQ plot. you can see it starts from here. It's basically and then towards the end it goes up. So that tells you that your analysis is good. So this is a genetic graph that compares the distribution of observed test statistics and the trait association, again, the expected. So the plots goes diagonally. So the point of inflection is of when the tails goes a little bit up. That's when you can see that your analysis is good. This graph here. Always when you see people publish when they do GWAS, that's the explanation. And then the Manhattan plot, basically you're plotting your log score, which is log of the odds, and then you can get the cutoff point. So those ones that are basically the SNPs that are contributing to the yield are the ones that you select. So on the x-axis you have these chromosomes numbers here, and then on the left you have the load score. Okay, those are how you interpret the Manhattan plot. So what we did here, those guys in Europe collected information on grain yield and other phenotypes, but we're doing only single trait. And then they compared the association with all other SNPs that they had. And then out of all these SNPs, this is the results here. The trait was grain yield, and this is the SNPs, and these SNPs are contained on these chromosomes here. This chromosome 6, this position of that chromosome, because maize has already been sequenced, and some of these SNPs, they know where they are. You have your allele frequencies and others, and then you have your SNP status here. You have a proportion of variance of the SNP. So how do you know that you did a good job? Of course, you get from the p values, and also from this graph here, quantile plot. So it starts in the beginning, and then towards the end, the tails go up, and this is the explanation. It's a genetic graph that compares the distribution of self-test statistics, your p-value of SNP association against the expected distribution by null hypothesis with no association, because that's your null. So the systemic inflation data looks good. The upper tail points above the diagonal that the true SNP is associated with what? With your trait. And our trait here is what? Is yield. And then how do you select your SNP? So you have to have your log of your odd, the log of your odd ratio. It's called log. So here we selected 6, you have these 2 SNPs here. But if you reduce maybe to 4, you get more SNPs and so forth. Okay? So this is the explanation here. The Manhattan plot explanation. So it's the x-axis is your genomic position of the SNP ordered by chromosomes. And the y-axis is your log p-value of the SNP of the association. So the horizontal line is your Bonferroni corrected, which is basically your fault discovery rate. So you see here we reduce from 6 to 4. We have more SNPs that we can work with. Okay. So you have to decide which threshold based on maybe literature or based on some experience where you're going to cut to make your cutoff. Okay, so we have two things to cover that we have to do. We have to do population structure and diversity. But before we do that, Peace is going to share with you post-survey. Let's take at least between 5 to 7 minutes and do that. Then we'll come back and do population structure and then genetic diversity. Okay, let's do that. Please go ahead and share with them. Thank you.
Stephen,
Yes.
I don't have permission to share to everyone, so I
Okay.
can only share to the host, so you need to try on your side.
Okay, let me try here. Okay.
So the link is on the chat. You can just get it from there.
In chat?
Yes, I
This
put it on the chat, but you can be able to see it.
or this one here. So you cannot share with the rest? Let me try.
No, no, I cannot. Um,
So if I share, will they be able to do it? Or I have to go back to the form? Let me first go back to the form here.
so I put the link on the chat. You can see it on the chat. You
But they
can
can't.
see it, but the rest may not be able to. So you can just copy the link from the chat.
Okay, let me do that. Okay, I copy again. I copy it back.
Yes, and send it to everyone else. Um,
Okay, let them try. Let them type one if they can be able to see and get access to. Okay, thank you.
now they can see it.
Okay, thank you. So I'm going
All
to give you
right.
at least 5 to 7 minutes so that we can finish the population structure and diversity before 5. Okay, thank you. Thank you.
Dr. Apia, people are saying they are done.
Okay, thanks for letting me know. So let's continue, okay? Okay, so we're going to talk about population structure. So what does population structure means? Basically, it's a bigger group made of smaller subgroups, which differ from family backgrounds, which can affect genetic study results. So what's the importance of population structure? So if population is subdivided, they can evolve uh, independently because then there's no breeding among the population. So by doing that, it allows population to diversify. So there are several methods of determining population structure. One is principal component analysis, structure analysis, and principal coordinate analysis. You can also do cluster analysis and others, okay? So in Ada Studio, we have a package called bioconductor called layer. So this package, it does population genetics, population genomics, landscape genomics, and genotype by environment. So it can also learn structure analysis. So it also can do genome-wide tests for local adaptation. Another thing that it can do is it can also do imputation of missing genotype data. So it include statistics method of estimating ancestry coefficients and so forth. Okay. So we already installed this. Okay. So in order to use this package, you used to change your format to genotype. So sometimes when people use this, if you don't change your format to genome, it won't work. Because most of the data that we get a VCF format. I mean, 99% of data that people get after SNP calling is a VCF format. So you have to change that. So here, there's a function called VCF to genome that's used for converting, okay? So we have in one of the files that we downloaded today is called data underscore pop, and that's a VCF file. So that VCF file we're going to use to do population structure. We're going to use principal component analysis, and we're also going to use admixture analysis. So always when you do population structure, you do most of the time you have to show this admixture structure. And the person who introduced that it's called Patterson. So the good thing with this package, you can do both population, principal component analysis, and also you can do administered structure. So what we're going to do, we're going to run uh, the script for population structure. It's going to give us our principal component, and it's going to give us our structure. And then I'll explain what that means, okay? So let's go back to our other studio, and we're going to run. I'm going to clear up this. I'm going to clear up this GWAS. Clear this up. So there's a file called population structure. Okay. So we're going to first run the first part. We're going to run this up to here. So if you can run up to let's line up to line 26. Line up to line 26. So if you can see my graph, this principal component graph, I'm going to explain something for you. So this population, if you look at, if you can see my graph type 1. Okay. So using principal component analysis, this population tells us that we have 1, 2, 3 population. So when you're using principal component, there's what they call skew plot. So where it bends and start being flat, that is before you reach there, that's what's telling you that the population that we have only 3 population. You see, there's 1 population, 2, and 3. Okay, then how can we prove that we have 3 population? Principal component told us that another method is called structural analysis. We're going to run that up to, we're going to run from 33 up to, let me see, up to, mm, up to 91. You run from 33 up to 91. So the principal component told us we have 3 population because before that's flat, where you start seeing flat points, data tells you that we have 3.
Hey. Oh, Dr. Opir had been out, but I think he's back now.
Stephen, you're muted.
Do you see my screen? Okay. Thank you. So we have 3 population. So these individuals here at the bottom, that means that the first one, so you can see that the DNA is distributed, the SNPs is distributed in all these populations here, in all these individuals. But the first one with all the green, so most of the SNPs are coming from this population here. So these individuals here belong to this population. So that's the first population. The second population, this on the left, you have ancestry proportion. So the proportion of SNPs that's coming from ancestry. The second population, you can have light blue. So most of their ancestry DNA is this one. And the third population is this one. So if you go back to the PCA gave a 3 population, not so. So if you go to PCA, these individuals here at the bottom, you'll find that they belong to this one here. And if you go to here, you find they belong here. If you go to here, they belong here. You can also run, we ran both 3 and 4 population. So this is 3. So let's see if the fourth one here. This is the fourth population. This one show 4. But you can see you have 1 almost yellow, 2 almost red, and then the light blue. But this small one is very, very tiny. So the principal component told us we have 3 population. And this one also, the structure, we confirm that we have 3 population, okay? Now, we can also go and run a plot. That plot can also, like a dot plot can also guide us to show that we have what 3 population, okay? So the last part that I'm going to rush through is genetic diversity. Let's see here. Where are my slides? So PCA, we've already explained this, population structure. Okay. I mean, this is what I was explaining. You can also plot this entropy. So when we are running that structure, it was giving us cross entropy. So the cross entropy you can plot is the lowest. So here you start with 0.78, and then as you come down, so this is the lowest here. So the lowest will give the number of population here. So that's why I ran the fourth one. We may say, okay, we have 3 or 4, but the 3 is the lowest part. So there are 3 methods. You can use principal component, you can do cross entropy, and you can do the structure. Okay, so those can guide you. Okay, so the population structure 3, this is 4. The last part is genetic diversity. So again, this is the process. You do the quality control, do alignment, get your SNPs, and you get your VCF file. Here we did our GWAS, we did population structure, the last part is genetic diversity. So what's genetic diversity? So genetics basically is related to traits passed from parents to offspring. And diversity is having a range of different things. So genetic diversity basically a range of different inherited traits within a species. So gene diversity is like having a large toolbox of genes. The more tools, the genes variation is the more you have. So the better chance you survive, like climate change and so forth. So take me back to the climate change, the maize. So the ones that survive, since that they had a different type of diversity. Okay. So what are the methods using genetic diversity? So we have genetic diversity, we have other methods. There's analysis of molecular variance. We have distance methods. You can do phylogenetic methods. You have fixation index, and you have polymorphism information content and others. So here I'm just going to show, I think, 1 or 2 of them. So there's a package called stamp. So that stamp also comes with it comes with the data. So this is done with potatoes. So the potato, there were 3 potatoes population. There was 30 samples and 46 genotypes. Okay. So when you convert the genotypes to allele frequency, so after converting that, then you can do the calculation for your genetic for the diversity. So the good thing with R, sometimes you have, if you cannot get data, you have some of these packages that don't really have the data, and you can use those data. Okay. So to use this package, you always use a distance called Ney genetic distance. I'm not going to go into details. So it's one of the most common methods that I use. So you use that to calculate the distance. And then after you calculate the distance, you calculate the distance between the populations, and also calculate the distance among the individuals. For example, we calculate the distance between population 1 and population 2. So distance between the population by itself is 0. So population 1, this 1 and 2, this is the distance between them. And 1 and 3, this is the distance between them. So the closer the distance, that means you don't have variation. So the further the distance, that means you have the variation, okay? Okay, so this genetics. So that was among the 3 populations. You can also do within the samples. So this is within samples. So this sample 1 by itself is 0. Sample 1 and sample 2, you can also get the genetic distance. So the closer the samples, the more the closer they are. Okay. Another method that's used for diversity is AMOVA, which is analysis of molecular variance. So it's similar to ANOVA, but of course you use the frequency. For example, here, instead of using like when you are in ANOVA, use the original values. Here you calculate the genetic distance. So this genetic distance here is now what you use for your ANOVA. For your AMOVA, but when you're doing the ANOVA, you're going to calculate the real the real information that we use for calculating the distance. Okay. So this is how you do the ANOVA. It gives you the degree of freedom of the population. For example, the degree of freedom of 2, the error comes from the samples and so forth, and then you get your p-values. Okay, so I was fast because I wanted to at least to make sure that I covered at least genomics. So I was fast, but you have the scripts. We're going to send you the notes. And we apologize that we went a little bit faster because today is our last day. So with that, I would like to thank the forum for giving us this opportunity. The last 3 months, we gave 4 modules, 4, and was 12 every module, and then you take another 2 weeks. So in total, we had 4 months. And then you, the attendees, we came to know you for the questions you asked, and you challenge us, and we learn from you. We hope to work with you again in other ways. And the particular data science group, a piece, me, and the people who are in the background. So thank you very much. With that, I'm going to give you back to Ruforum so that at least give them some time. Thank you very much.
Thank you so much, Dr. Opio. We are so grateful for this long training, and it has been beneficial to us, also to the participants. And we thank all the participants for being part of this training. And I encourage all the participants, if you want to refresh your memories on the training, you can always visit our YouTube channel, Ruforum Network, for revision and better understanding. And also, we are in the WhatsApp group together with Dr. Opio and Peace. So if you have any questions, you should be able to just get back to us, and we'll be able to help you. Thank you so much, and have a good weekend. Bye.
Bye.
Oh, thank you so much.
Richard, yeah, this was tough.
Yeah.